Give a brief introduction on Abhi. Abhi is least well known as my previous office mate and much more well known for many other things. He did his PhD at CMU in the Mohimani lab, working on antimicrobial identification, and then made a switch to core machine learning, and most recently has been at the Arc Institute and came out with this really, really interesting model state. So Abhi's been a lot of places and done a lot of things and has done it all very, very competently. So we're super excited to have Abhi here today to talk to us about state.
Awesome. Thanks a lot, Caleb, for the kind intro. I'll just go ahead and start sharing my screen. Just 1 second. There we
And so while Abhi's getting set up, for folks who are just joining, you can feel free to put questions in the chat, and Shaheen and I will moderate so that Abhi can focus on giving you the best presentation possible.
Yeah, awesome. And then for you guys, please feel free to just hop in with questions. Happy to keep it interactive. So can you see my screen?
Yes, yes, again.
Okay, perfect. Awesome. All right, thanks, guys. So today I'll be talking about our preprint on predicting cellular responses to perturbation across diverse contexts with state. And so I'm a ML scientist in the ML group at the Computational Tech Center at ARC. Before I get started, just a huge thanks to everyone on the team that contributed. Folks from various groups at ARC, different people in different tech centers, and then also core labs and the leadership. So please check out the paper for a comprehensive list of acknowledgements. And then a brief plug before I get started. So ARC recently put out its virtual cell challenge. And we have a couple of resources here to help people kind of get up and started. A blog post kind of describing for more data science or ML centric folks, like what is the problem of perturbation prediction? And why is it not like a super well-defined machine learning problem? And how are people currently setting it up? And we also have a collab notebook for people to kind of get started with hacking on that. So let's get started. So understanding single cell responses to perturbation is something that would be really helpful for therapeutics development. It would offer insights into biological mechanisms. If a simulator actually worked, say for the genetic perturbation case, you could simulate what the effect of a genetic knockout would do. And that would help us understand what genes are implicated with other genes, what pathways those might affect, and then how do we drug those pathways, right? For the drug perturbation case, it would help us understand what drugs may help reverse cell disease phenotypes. And the most important part here is that it's more scalable than wet lab approaches. So you can test something out in the simulator, see and use that to guide your more expensive wet lab generations. And in general, this feedback loop gets faster once you can start to reliably simulate cellular responses. And so there's been many attempts at doing this decades ago, and these were more traditional systems biology approaches that tried to keep track of all the different processes in the cell and take a more systems biology approach to modeling these in different interactions and different processes. But these approaches struggle to scale is the fundamental issue. And it's just because like in the eukaryotic cell, there are just enormous complexities as compared to more simple cellular models that this has been done for. But such a model for the eukaryotic cell would be super useful, right? So if you have a given initial cell population, say that it's phenotypically diseased or maybe phenotypically old, it would be really useful to try to figure out what different ways can you poke at that cell to move that cell population to be more phenotypically healthy or more phenotypically young looking. And so a successful cell simulator model should be able to identify how do you perturb the cell to induce desired phenotypes. And so throughout the talk, I'll refer to these cells as these little histograms. These are just distributions of gene expression. So like here, maybe gene 3 is highly expressed, but gene 1 is lowly expressed. It's a common featureization of the cell. People have their problems with it, but one thing about it is that it's starting to become a scalable featureization of the cell. And so people can do what are called perturbed seq screens. Here, they basically pair each cell with a different perturbation ID, so to speak. So I'm kind of converting this into computer science speak right now, but the idea is that when you get these readouts from the cell, these are mRNAs that you're reading out, you know what perturbation was applied to that cell, and then you also can align, the read itself to identify what gene was being expressed. And so you can build out these histograms for each of these cells at single cell resolution. And the scale of these data sets are also rapidly growing. So recently, there's been a bunch of new data sets that have been put out. Zyra Therapeutics put out a 10 million genome-wide genetic perturbation data set. So that's kind of the first of its kind, where it's perturbing almost, I think, 19,000 different genes. Parse PBMCs put out a cytokine signaling perturbation data set. This is a really high quality data set as well, with 10 million cells and only 90 signaling perturbations. So a lot of depth for each of those perturbations. And then the Tahoe 100 million data set put out 100 million cells. Here, it's about like 1,100 different drug perturbations. And the data is a little bit more sparse in terms of UMIs per cell, but here you can just get a massive amount of context and different cell lines, both ranging from disease cell lines to healthy cell lines. So as I'll get into a little bit later, it's useful for the task of state, which is context generalization. And so machine learning systems have been seen in the past to be able to learn complex biological systems. AlphaFold, for example, is able to map protein sequence to structure, and it's now used by scientists to rapidly assess the effects of mutations and simulate biomolecular interactions. And in fact, people can use AlphaFold in a similar style to like a perturbation model, where you can induce a change in the sequence and then see what is the resulting change in the structure. And so it enables this sort of counterfactual analysis. AlphaFold was a step function improvement also at the time in terms of structure prediction. And one of my favorite quotes from Nobel laureate John Jumper is that AlphaFold 2 was largely the same training data as AlphaFold 1, but just better machine learning algorithms that better match the inductive biases of the data. And so what about for perturbation response prediction? AlphaFold has shown that machine learning can learn instance level functions where you can map a particular protein sequence to a particular structure, kind of simplifying over protein conformations for a second. But single cell perturbation response data are not instance level. In fact, by measuring the cell at single cell resolution, you actually kill the cell. And so you don't have causal training data in that you can never observe the same cell before perturbation and then after perturbation. And so the correct level of granularity to think about this is you have a distribution of cells that was under some, DMSO or control population, and then you have a distribution of cells under perturbation. There's other things that also further complicate this than just the learning task. So single cell data is notoriously noisy and has a lot of technical artifacts. So if you do the same experiment using like different sequencing technologies, that can actually give you different biological results, which is, a problem because if you're trying to probe at true underlying biology, right? And there's a lot of unannotated conflators in the data as well. So biological heterogeneity, like cell cycle within a given distribution of cells. And a hallmark sign that, the machine learning here is not maybe has something to be improved is that even with significantly more data, methods have not shown proportional gains. And most are actually outperformed by simple mean baselines. And so we would argue that we're actually not quite yet in the regime of scaling and machine learning if you can't see better performance with better data or more data. So we wanted to revisit what is the data generating process and design the method with those inductive biases in mind. And so the way we kind of scope it out in the paper is you start with some cell state A, and can you guys see my mouse?
Yes.
Okay, perfect. So you start with some cell state A. Here we're calling this distribution D basal. And say you induce some perturbation on it. It could be a CRISPR-I knockout, some drug perturbation, and this gives you a new distribution XP. And we say that this is comprised of three different effects. So one is some true perturbation effect that the knockout would have on the cell. There's an additional factor of biological heterogeneity. So different cells within a given population can, say, be in different phases of the cell cycle, and they might react differently because of different accessibility patterns at those times or different environments, right? And then lastly, there's some readout noise associated with the assay itself. And so this is kind of like in machine learning speak, irreducible noise. And we represent each of these terms as TP of D basal as the true perturbation effect on the basal population. And then H of D basal, this is the biological heterogeneity term. And then lastly, the technical noise term. So this epsilon here defines the data generating process for the perturbed distribution.
Can you talk a little bit here about the, what it implies that this is an additive model of these different noises? So like is intuitively, right, cell cycle is something that may change in response to perturbation, right? But under this model, these are explicitly disentangled. And then I'm wondering, when you do learn this uh, distribution of biological heterogeneity, what does it actually, I mean, what does it actually seem to learn? Is there any, yeah.
Yeah, so for the first question, it's a little bit of an abusive notation, and the actual quantity we learn is just a transformation on D basal, and it's kind of been separated out, maybe slightly an abusive notation into an additive model. So it's more for illustrative purposes, but like the transformer that you train is actually just a black box function on D basal. For the latter function, I think it would be a really interesting research problem to probe at what kind of things it might have learned about H of D basal. And there are some ways that you can get at that. So like autoaggressively feeding the model generations back into the input of state and not applying any perturbation, just like the control perturbation, and seeing if that would model some of the heterogeneity, or like if you can, build something that can predict cell cycle from a given transcriptomic profile, see how that changes.
Did you try this? That's sort of an interesting, like, yeah, you did. Yeah. How did this go?
No, 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 we didn't. Yeah, so I think it's a
Ah,
super interesting next experiment, or
okay.
something fun to try. Yeah, yeah. But it's a good point that it's not truly an additive model that it's learning. Okay.
But additive in terms of like separation of concerns. Yeah, maybe you can talk a little bit about this as you get into the model architecture, right? Is the model actually structured this way, or is this well, because I think it's um convenient to have like the modeling primitives in place and say like these two are disconnected in terms of their data generating process, even though they are both related to the outcome that we're observing, right? And that seems to be the way that state is structured, but uh, maybe I'm misunderstanding like abusive notation compared to like the model architecture. But yeah, thanks.
Yeah, yeah. So I think it's a good point. Maybe I'll first describe what state does, and then I'll describe what a true additive model would do and why state is maybe not a true additive model. So we broke state up into two different models. And the first model, we call it the state transition model. This models the first term of like the true perturbation effect and then cell population heterogeneity. And it's a transformer model, so it operates between feed-forward and self-attention operations across, we call them sets of cells. So here you take some subsample of your entire control distribution, and you take its transcriptomic profile. So these are just gene expression count values. Specifically, we log transform and then just use those values. And then we also append a perturbation token. And so for a given mini batch, a given set, you're going to have all of the cells applying the same perturbation to that set of cells. And to reduce the like one of the reasons that we do that actually is to reduce the learning burden on the model. We tried this approach where we did not sort by known covariates and kind of just threw cells with different perturbation tokens within the same set and hoped that the model would figure it out. But it turns out that perturb-seq data is just too noisy to do that. And so when you sort the cells by covariate for each given mini batch and set, making sure that all of the cells here are learning the same perturbation effect, it seems to like drastically change the learning dynamics there. And ultimately, the model no longer has to deconvolve the cell type differences to perturbation from the perturbation effect. And so maybe the difference from a true additive model here would be if you learn something with this perturbation token, and then you have a separate transformer that learns H of D basal with no perturbation token at all. And then you add the effects of those two models. So one would be like a reconstruction model, and one would be like a perturbation model. In this case, it's like kind of all convolved into the model. And the only sort of way you would deconvolve anything is through the sorting process. So like we sort by cell type and sort by perturbation. And so those are things that are no longer convolved within a set of cells. But H of D basal and like cell phase and things like that are still convolved in the model. Those necessarily have to be because those are actually unannotated in most data sets.
Interesting. I wonder if you'll be able to comment on how self-supervised learning seems to respond to this type of noise, because I think it seems like in other regimes, like self-supervised learning seems to perform particularly poorly on noisy data. And extending this to the setup here, maybe it's the case that our perturbation readouts are just not, viable cell states for the most part, or not healthy, or for some reason they're just more noisy. I'm not sure if this would be the right way to interpret it, but it's interesting that the model seems to be like fine, or at least as fine as it can be, when it doesn't have any context or metadata for the basal states or the biological heterogeneity, because we just don't have this. But it made more sense to separate out the perturbation conditioning for another day, right?
Yeah, definitely. I think it's a difficult problem for sure. I think this is quite not in the regime of self-supervised learning, so I can't comment on specifically state transition for that. I see what people are doing with like the DNA stuff where you're also like very noisy and it seems like it is more difficult there. I would say the main difference or the main difficulty here is just the huge amount of noise. And at least in other regimes where people would typically do self-supervised learning, um, a lot of times they can do cross data set learning because the noise comes from the underlying data generating process and not so much like the technical readout of like DNA sequence, for example. Of course, there is some error in technical readout of DNA sequence, but I would argue that perturb-seq is much more noisy and prone to batch effects. And so it's been traditionally pretty difficult to train large models across huge data sets in self-supervised fashions.
I see. Okay, and I do want to let you get on, but we do have one relevant question for this section here, which is, can you explain more about the covariate matching strategy? Which covariates, what are the trade-offs for rare exemplars of certain categorical covariates?
Yeah, great question. So the concrete trade-off is the number of cells that group under the same set of covariates. So one covariate that we always group by is cell type. What that means is that all of the cells within a given set going into the transformer are the same cell type. And that's because we want to incentivize the model to learn cell type specific effects of perturbation. Another one that Caleb mentioned is that we're grouping by perturbation, right? So we want the model to more easily learn perturbation effect. And so each individual set, we apply the same perturbation token for everything. Another one could be like the batch or like in uh, the technical batch, right? Like the experiment number, the well plate. All of these things can also be sorted by, and you can make it such that your individual set is all cells from the same well group, for example. And so we tried this. In our code, we call this like the batch map as opposed to like the random map. And it has an interesting modeling trade-off in that when you filter for this in, say, like the existing genetic data sets, genetic perturbation data sets, it ends up being a really stringent filter. And so you can only get 2 to 3 to 4 cells sometimes for a given cell set that match on all of those covariates. And this empirically seems to make it harder for the model to actually learn to denoise the perturb-seq. And so there's a trade-off between the cell set size and how stringent you're doing the covariate matching. And we actually found that data set by data set, if you did this batch filtering, it would have different effects. And so in our case, we weren't able to find convincing evidence that grouping by like well group and batch effect was actually helpful. So we decided to just kind of offload that burden to the model. It's a really interesting modeling trade-off though, and I think people are going to have to investigate it more and try out, what is the best sorting by strategy for given data sets, or is there one that is universally better across most data sets?
So I also have a question. Do you treat covariates that do have pure population with no let's say, for example, when we are talking about batch, there is no pure population that doesn't have any batch effect. But for some of the effects, you might be able to estimate it from a control population such that, oh, I can model what would be the effect without, and then remove it from the data. And that's the key that we couldn't do in batch correction because we don't have a way to estimate it. But some of the covariates in here might be, for example, again, the perturbation effect. We do have controls that don't have a perturbation effect, so you can estimate your basal that you talked about, the basal variation, only using the ones that are, non-targeting or other control ones. Have you tried any of those approaches to estimate basically the, let's say, either in this case H that you're referring to, or equivalent versions?
Yeah, it's interesting. We have not tried that. So I guess what you're saying is you can infer different sorts of I guess properties about these things and then use those in this process. We haven't done that. The only sort of basal thing that we look at is the perturbation.
Yeah, thank you. And again, the one I was mentioning specifically because we have two type of controls. Again, if it's perturbed, we have non-targeting and we have no guide cells that didn't have any guide. And based on the differences, you can estimate that basal level of variation that you expect in your control population. That's what I was referring to. If you can estimate it directly before and then injecting it here.
Yeah, makes sense. And maybe it's worth pointing out that in order to actually properly learn basal variation, like one thing that we do in the model is even for non-targeting cells, we treat that as a perturbation. And so what that basically means is you learn to transform a given subsample of non-targeting cells to a different subsample of non-targeting cells. And empirically, that seems to be doing better than just asking the model to learn the identity function of like predict the exact same subsample of cells.
Makes sense.
So, I mean, kind of some evidence that hopefully the model is learning some notion of basal heterogeneity. Awesome. So just recapping the learning problem then before I move on. Concretely, the inputs to the model here are some batch dimension by the cell set size. This is a chosen hyperparameter by the number of genes that you want to model. Typically, we just choose like 2,000 highly varying genes for the preprint across the different data sets. And the output is the exact same dimensionality-wise. It's just now these are after applying the perturbation. And so the state embedding model captures the last term, epsilon, which is more so dealing with the measurement readout noise and technical artifacts of the perturb-seq datasets. So state embedding is specifically trained across many different datasets. It's an encoder-decoder model that I'll kind of dive into the architecture a little bit later. But the learning task here is to predict gene expression variability. And specifically, it's a bottleneck representation learning task. So the goal is to learn a single cell embedding and then decode out gene expression from that single cell embedding. And the learning task that we design here kind of encourages the model to capture subtle phenotypic variations across different treatment groups. And training it across the data sets help minimizes data set specific noise. And so you can plug this model as an alternative featurization for each cell into the state transition model. And then if you do that, you also train like a decoder back into gene space so that you can like kind of interpret it more biologically. And so before I jump into the results of the paper, I wanted to just provide some intuition on why this approach might work better. So it turns out that state can be shown to generalize a lot of common approaches in the field. And so pretty trivially, state can learn a suitable perturbation response model where self-attention is the same as mean pooling or averaging. And you'll see this happen in some of the attention heads when you train the model. It often learns these vertical lines for the keys, showing that it's attending to each cell kind of the same. And it can also learn single cell responses. So similar to like repurposing an SCVI model for perturbation prediction or for gears. And in this, you would map an individual cell from the perturbed population to an individual cell in the control population. So a random one-to-one mapping instead of cell sets. And you'll find these in the diagonal attention matrices heads. And it can also learn some complex distributional effects. We think most likely this is probably just learning some denoising effects. And you'll see these in like the attention matrices with more complicated distributional patterns. And there's also a result in the paper. Beatrice and Mingze and some others showed that under some regularity conditions that are that both the rows and the columns of the attention matrices sum to 1, that the continuous optimal transport map between these population actually lies within the state solution family. There is one more caveat there, that the cell set size has to go to infinity. So maybe it's not actually learning the OT map, but it is a class of solutions that can be a generalization of OT-based approaches. And I would imagine that in some cases, if it's beneficial for the model's task, it probably learns something similar to the OT map. But this isn't something that we've dig so deeply into in the preprint. Next, we wanted to convince ourselves that these cell sets were actually doing something. So we ran an ablation of keeping everything else the same, including the number of cells through each forward pass, so increasing the batch size as we decrease the cell set size. And we just wanted to see, how does the validation loss change as a result of changing the cell set size? So when you have cell set size 1, I think the model just gets confused. It's like too many different perturbations all at once, so it doesn't really learn. But then pretty dramatically, as you increase the cell set size, the model gets better and better at reconstructing perturbation transcriptomes. And this happens up to a point. So like cell set size 256 seems to be doing really well in terms of validation loss. But cell set size 512 starts to do a little bit more poorly again. And our hypothesis here is that this is dataset specific. So this is on the Tahoe dataset, but on the parse PBMC's dataset, which is much deeper per perturbation, you actually find that cell set size 512 does better than cell set size 256. And so most likely in the case of this plot, the 512 is kind of just approaching a pseudobulk model to a degree, and that there's probably an optimal point that should be ablated for different data sets. And then just to really convince ourselves, we also compared to a pseudobulk model that explicitly replaces the self-attention with just an averaging operation. And then we also compared against state transition without self-attention. So this now just becomes a feed-forward model, but with fancy, batching and group by for the covariates. And in both cases, that also seems to perform much more poorly in terms of validation loss.
So, Abhi, can you, this is going to be a stupid question. Can you explain to me why pseudobulk is non-optimal? Because without paired samples, it would seem like pseudobulk is the best you could possibly do.
So pseudobulk it's a hard question to answer, right? Because it's essentially asking like, what is the model attending to? And that is a level of interp that we haven't had a chance to do yet. But to me, intuitively from training like a lot of these models, it seems like the pseudobulking helps because it denoises perturb-seq. And my intuitive answer is that self-attention helps you better denoise perturb-seq. So like maybe it looks at a cell and it says this is a bad quality cell, and like I shouldn't let that bias my pseudobulk, for example. It just seems like letting the model look at a larger subsample of the cells before having to make a prediction kind of helps it denoise.
Okay. Okay. Thanks.
Yeah. So it's sort of an unsatisfying answer, but we want to do a little bit more interp work on it and like really dig into why, because it's an interesting question, right? So then we wanted to move beyond just validation loss, MSE, and some other common approaches in the field that are a little bit more machine learning inspired. So we introduced, several people on the team introduced Cell-Eval. And here it kind of hits on 3 different types of metrics. One is just expression counts, and it deals with expression space. The second is once you go from expression space to like differential expression analysis space, right? So like looking at log-fold changes of specific genes and such. And then lastly, we also looked at the effect size of the perturbation. This is just how many genes were significantly changed in response to the perturbation. I'll go into some of these one by one. But first, the learning task. And so I alluded to it earlier, but state doesn't actually do perturbation generalization like gears and some other models in the field. State is really focused on context generalization. And so the best way to think about that is in terms of this kind of diagram here. For a given perturbation that you have seen in a different biological context, so this is a different cell line of the same data set, or even a different data set altogether. You want to learn the effect of this perturbation in a new context for which that perturbation is unseen. And so in the paper, in the test context, we look at 30% of the perturbations randomly, and then we evaluate on 70% of perturbations that were unseen in that cell type. And then two really strong baselines that we compare to in this field are the perturbed mean. This is just if you average the perturbation effect vector across all of the biological contexts in the training data, and then apply that same delta to the control population in the test set. And then the context mean, this just averages the transcriptomes of the seen perturbations, so this 30%, within the test context. The idea for these two perturbations is firstly, I mean, it's really difficult to actually beat both of them with deep learning approaches. And secondly, if you improve on both of these, then by definition, you're learning some cell type specific effects of that perturbation. And so this was kind of the motivation for using two baselines.
Did you ever happen to try a baseline of the context mean, but not mean in terms of like the closest cell type in terms of like L2 distance from the query cell type?
Sorry, I was just swatting away a fly. No, we haven't. I think the nearest neighbor baseline is also similar in performance, but it's not in the paper.
Okay.
No. So going through metrics, first one that we looked at was actually introduced by the perturb bench paper from the Altos Lab folks. We adapted we adapted it a little bit. So what we did was we took our prediction, and in terms of the Manhattan distance or the L1 distance of the transcriptomes, we ranked what are the true perturbed profiles in the training data. And then the score that the model receives is the rank of the true perturbation to our prediction for that perturbation. The reason we use Manhattan distance is that the distributions of these discrimination scores are really biased if you use cosine similarity, actually. And I think there is some work showing that cosine similarity is oddly behaved in some circumstances. So we found that L1 distance seemed to be the most well-behaved across all data sets. And then we just renormalized the score from 0 to 1. And so the idea is if your model has a really high perturbation discrimination score, it's learning different effects of those perturbations, and it's able to, if the ideal goal is to build a model that will tell you what perturbation to apply to get to it in like desired phenotype, you would want to do well on this. And so here we see that, I guess I should first go through the colors here. So the blue bar represents state. And the yellow bars across all of these plots now are going to represent the two different means. And then we looked at a couple of different common approaches. So a linear baseline that we re-implemented that was recently published, SCVI, where we repurposed the batch effect into a perturbation token, and CPA and SCGPT as well. We see that on the genetic perturbation data set, we're about equal to the perturbed mean baseline and significantly better than some of the other simpler or deep learning approaches. Where we start to pull away from the other methods is in the signaling perturbation data set, where we see a 29% increase over the next best performing model, which in this case is actually SCVI and not one of the means. And then on the Tahoe perturbation data set, this is where you have the most number of contexts and the most number of cells. There's a really clear benefit of using state over any of the existing both simple and deep learning based approaches and the strong mean baselines.
So,
And this is a recurring theme that we'll see. So like as the data set scale increases, the relative improvement of scale also increases.
so good question regarding if you split this performance, because again, perturbed mean is gonna be well if it is a housekeeping gene that has similar effect across cell lines. If I recall correctly, it's the same one in different contexts, right?
Yeah.
Same perturbations. So have you split that effect by gene class? That is to say, for genes or perturbations in general that has, let's say, similar cell type, like affect cross cell types. Let's say if it's gene knockout, if it's a housekeeping core process, let's say I'm knocking out parts of the ribosomal complex, it's expected to have like at least comparable signal, whereas some, cell type specific signaling components might be much different in behavior. So have you done any splitting on seeing which category of genes or perturbations or better predicted in that case with state compared to, because again, state tries to learn cell type specific effects, so it may even perform better for those sets that are not ubiquitous.
Yeah, so just rephrasing, so tell me if I caught that correctly. You're suggesting that it may be better to average the perturbation effect selectively over biological contexts, depending on like how similar those contexts are to the test context. Is that correct?
So basically here you're averaging across all perturbation in the plot that you're showing. What I was thinking in the results that you were showing. I was saying that you can split that by perturbation class. That is to say, for, let's say, ubiquitous perturbations that are expected to behave similarly across different cell types, perturb mean might be actually very good, right? Because we don't expect cell type specific or context specific changes. These are core processes that are the same. Whereas for processes that are very cell type specific, you may observe that if I split the perturbations into housekeeping or ubiquitous versus the ones that are cell type specific, I may even see more gain by using state because now I'm like focusing on those cell type specific perturbations and I'm not mixing them with ubiquitous ones.
Yeah, it's very possible, and it's not something that we split them for. I think if you look at where does perturb mean do well, and you looked at that, my strong suspicion would be that it does well on like housekeeping genes and other such ones.
Yeah.
It would be interesting to see if we split what the improvement would be.
Yeah. Likely for the cell type specific ones, you might see even more difference in the perturbative data that doesn't seem to have that difference yet as much.
Yeah.
No, Abhi, I
So the
want
next met
a
yeah.
question here because you mentioned the scale of these data sets seems to correlate with the relative performance, right? Um, and you mentioned doing this, I think, sort of 30% split on the test conditions, right? So this also means that for the larger data sets, the model is essentially seeing more of the test time context, right? So do you think that this relative performance is an artifact of that? Because also for an approach based on transfer learning, you would sort of hope that the most pronounced improvement is actually in the limited data regime, right? And what you're trying to say is if I have a virtual cell, this is how much data I need to gather in order to get a reasonable predictor for unseen conditions on this, this test set, my cell type.
Yeah, that's actually an interesting point. So all models would be seeing more of the test context. So I think state having a relative improvement makes sense, but it's a really good point you bring up of you are actually seeing more raw number of cells. And so like in the inference application of the model, these results aren't suggesting like just from these results, you can't conclude that, you can just take the same number of cells as you would in a replogable data set and see the same improvements over the mean baseline in that inference setting.
I guess it is sort of we've actually talked about this offline, but it's even if there's not an explicit scaling law study, I think this does sort of hint at the idea that if you were to collect more data, it would improve the model more, which is, it's sort of a basic question, but an important one to answer for a transfer learning approach, right? And you can at least, am I understanding that this is actually what's being shown across data sets here? Like the bigger the data set, the more benefit to relative performance. So if you can collect more data, it will improve your virtual cell.
Yeah, definitely. Yeah. And I think at the very least, like you will always, in most, like to my knowledge, you will always have access to some control cells of like you would collect some cells and then you would want to analyze those cells, right? But like the question is how many. So yeah, a proper scaling law would be interesting for that and kind of showing even like something to show that would be varying the number of perturbations that you hold out instead of just a fixed 30%, right? And that would kind of probe at what you're saying, I think.
Yeah, interesting. Okay, cool. Thanks.
I just wanted to add one quick other note before we go. The same thing I mentioned, you can see it in this plot. The one all the way to the right, all the perturbations that you have, those 600 perturbations, 1600 perturbations are all housekeeping. Those are all essential genes. And that's where you don't get enough difference from perturbed mean versus the state, because these are housekeeping, so even if it's in a different cell line, you expect similar behavior. Whereas the largest that you see is in the parse data, that those are all cell type specific signaling elements and cytokines that you have. So the more specific cell type specific your perturbations is, that's what you see the differences become more pronounced. Again, like you can see it even in the plot in this case as well. Just wanted to emphasize.
Yeah, no, that's a good point. Yeah. And I think this is captured somewhat in sometimes in the field. I'm seeing more and more, like there's more and more papers out now where people are focusing only on top differentially expressed genes. And maybe the correct question to ask is like, how about for cell type specific effects of genes, and how do you subset analysis to that? But it's also something to be said about it's important to capture the biology of housekeeping genes as well. But yeah.
So there's one question I think makes sense to touch on here. I do want to let you
Yeah.
move, but there's a question that says, are these baselines sufficient? Many of these are no longer considered state of the art. And so maybe you can comment on whether this is, an artifact of the learning regime being different from some of these other models, or if it's, how you selected these baselines.
Yeah, so do you mean the deep learning based ones, or does the question refer to the mean baselines?
I think both, but maybe you can just comment on the overall benchmarking strategy. Personally, I think mean baselines, should always be there, and these aren't normally, and so these actually seem to be quite strong in this case, right? But
So
also,
there's two, yeah, yeah.
yeah.
So there's two reasons for the mean baselines. One is if you improve over both perturbed mean and context mean, then you can start to probe for cell type specific effects, just kind of by design of what those baselines are, right? The second is that they're just like really strong. So like you've worked in the field, and so I know you know, but a lot of people underestimate how powerful the mean baselines are, or maybe a better way to put it is how difficult it is to train deep learning models successfully on perturb-seq data. The other baselines, we do want to add more. There's been a lot of really cool work lately on different kinds of perturbation models. So it's something we're working on.
Okay, great.
Right?
Moving forward, there's one question about whether the model can predict perturbations on unseen small molecules. And if not, is the Tahoe prediction only holding out cell lines rather than perturbations? And then how do you evaluate this fairly?
Yeah, so the Tahoe data, again, it's holding out perturbations that were, it's holding out some perturbations and evaluating it on test contexts. And so you've seen this perturbation before, like say one drug of the around 1200, but you just haven't seen it in the test context. And we have 5 different test contexts. We don't right now generalize to unseen drugs. I think it's a very difficult problem to generalize to unseen drugs, especially given Tahoe, you have 1200 drugs and chemical space is a lot more than 1200 compounds. And so meaningfully generalizing from 1200 compounds is a large task. I think that data that has significantly more perturbations is maybe the first step, but there are some trivial things you can do, right? Like you can use Smiles embeddings, for example, or not Smiles embeddings, but Morgan fingerprints of Smiles strings. And that gives you a trivial way to generalize to unseen drug contexts. But it's not something that we put in the paper because it doesn't meaningfully improve performance. So I would argue that at least for the Tahoe data set, it's not the correct regime of data to tackle that problem just yet. And a lot of times when you're like setting up ML problems, you need data to meaningfully separate different ML architectures and see what works and see what doesn't. And I don't think we're quite there yet with the data for that problem. Right. So the next one's easy. We don't have to spend too much time on it. It's just Pearson correlation of the delta of the transcriptome. So the ground truth minus the control population. This is done on pseudobulks, and the predicted minus the control population. So again, here, this time we outperform the perturbed mean on the Replogle data set, and we match the context mean and beat out other methods. On the signaling and chemical perturbation data sets, we again see a larger separation of state over the other methods. And so it's promising to see that like now we're starting to see the first signs of improving with data scale. And this is like where you want to be as a machine learning practitioner. Probably the final metric that I'll go into in detail is our effect size metric. So here for the control population, we compare it against the predicted population and the observed populations. And for each, we run a differential expression analysis test and just identify what are the number of predicted differentially expressed genes and what are the number of observed differentially expressed genes. And we rank order the perturbations according to these numbers. So the effect size here is how many differentially expressed genes. And then we just report the Spearman rank correlation between predicted and observed for these two. And so we find that the model is actually able to learn effect size really well. It seems to be like a learnable characteristic of perturb-seq is effect size, even in like the replogal genetic perturbation data set. So here we outperform the means. And again, always good to see when SCVI does well because it seems to suggest there's something learnable about that metric in the data, as well as state. And then for the signaling and chemical perturbations, you again see pretty good separation from the mean baselines and other methods. One last metric that I'll briefly touch on is that there are other ways you can probe at what is the model learning, where you do see a huge separation from other methods on the genetic perturbation data. So here we report an area under the PR curve. This PR curve is constructed using p-values. So what we are actually varying when plotting out that precision recall curve is the score threshold on the predicted FDR. And so you're basically changing the cutoff of what is the score threshold by which I'm going to consider a gene significant or not. And then what we are reporting is the overlap with the ground truth under that varying score threshold for FDR. And so when you do this, you see that state perturbations are better calibrated in terms of the confidence of that prediction, kind of holding up across different significance thresholds as compared to other baselines. And the other data sets actually you see is small, like a little bit smaller of a gain as compared to on the replogal data set, but across all of these, we seem to do reasonably well. And this is if you actually plot out those curves. So you'll see that a lot of these at recall 100%, it's just recovering some probably housekeeping genes that are differentially expressed. So that's where you're getting this like 20% number, but state seems to improve across varying recall thresholds. So the error bar here represents different cell lines, and we basically did cross-validation across different cell lines to leave one cell line out. So I think as we've alluded to, it's nice that you're able to do this approach in the few-shot setting, but ultimately what would be more useful for biologists on the inference side of things is the zero-shot setting. So when you're not seeing any perturbations in the test context and you just have a set of control cells, can you predict how different, CRISPR knockouts or different drugs would affect that new cell population? So when we tried the same setup, we had a lot of mixed results. On some data sets, it actually works reasonably well. And on some of them, it was a little bit more challenging. What that suggested to us is that, okay, there's just a lot of batch effects when you train across data sets. And so how do you tackle that problem? And so that's where we bring in the state embedding model. And so state embedding is a encoder-decoder model that is trained across several different mega atlases of single cells. So we use ARC's SC base count data set, some subset of the Tahoe 100 million that's always kept in the training data. This doesn't really affect the previous results since those were on highly varying gene representations of cells, but we decided to include some of them in the state embedding model as well, just because there's so many of them. And the cell by gene data set from CZ. And so to train this model, it's trained to learn a specific vector representation of a cell. So this is just a dense embedding of the cell. And then it uses that dense embedding to decode out gene expression for other for various query genes. And so you should think about this as a sort of autoencoder. It's learning to compress the transcriptomic information of the cell into a single dense vector and then use that, like it's incentivized to put information into that to be able to meaningfully decode gene expression. And so that's what I just said here. So we'll go through this one by one. First, you start from just the gene expression counts from the given data set. For each of these, we take the ESM embedding of the gene and the log expression of that. We actually use SC Foundation's soft binning approach to convert that scalar value into the embedding dimension size. When I say ESM embedding of the gene, what I actually mean is the ESM embedding of all protein coding transcripts of that gene. And then we average the per amino acid embeddings, and then that gives you a transcript embedding, and then we average the transcript embeddings to get a gene embedding. Here we use 2048 as the sentence or set size of these genes that we pass into the transformer. We ablated higher set sizes as well, but found diminishing returns that didn't seem to justify the flops. And then we also append a learnable dataset token. So this is a learnable parameter that goes into the model. And we prepend a learnable CLS token. The model learns through this transformer to transform this CLS token into a state token or a cell embedding token, and transform this dataset token into, I guess here I've represented it as D, but just it's a dataset token. And these are trained with different tasks. So the dataset token here is trained to predict the dataset of origin. This is just a simple MLP, and it becomes around a 15,000 way classification task. What was interesting here, anecdotally, because SC base count is separated by SRX, we found that the dataset token actually learns a lot of cell type specific information in addition to correcting for batch effects. And that's because a lot of the SRXs in SC base count are cell type specific or cell line specific. So that was something kind of cool. And so this sort of way of introducing supervised information into the model is not novel. I think like scprint does something similar with different annotations. So it's a pretty neat way to get the model to learn this on its own, because this dataset token that goes in is a learned quantity. So the state token here, which is transformed from the CLS token, this is then passed to another MLP. This just predicts the log count or expression given the ESM gene embedding. And this is actually, it shouldn't be a vector. This is just a scalar quantity of read depth, since that's kind of independent. That's like an experimental factor and not like a biological factor. And then lastly, we also take a projection of this dataset token in here, kind of letting the model have some idea of like batch effect and such. So although it wasn't trained for this task, we see that on common integration tasks, it's able to do well and match models like UCE. So here we've plotted on the tabula sapiens data, different spleen cells, and you'll see that the UMAPs of these cells separate by cell type under both state and UCE. And they don't separate by donor as well, which is kind of what you want. You want to minimize donor-specific effects separating the cells and maximize cell type specific effects that are captured. The point of this is not really to say that state is better or worse than UCE at integration tasks per se, but it's just a task that we never trained for, and we still seem to do well, even though UCE was specifically trained on this data and state was not trained on tabula sapiens. So we can plug in state embedding and replace the highly varying gene representations of each of these cells. And so now we're just using this dense representation for each cell and prepending the perturbation token to those and passing it through the model. And then we train a separate decoder MLP to go back from the embedding to transcriptome. And so these are all trained end to end, except the embedding model. These embeddings are frozen. So we find that SE improves in perturbation modeling over existing foundation models. So this plot is a little dense. What we wanted to do here is see how well do different embedding spaces separate cells by perturbation label. And so what I mean by that is you take the training data and you train a classifier. Given a cell, can you predict what perturbation was applied to that cell? And if you can train an embedding model that can really easily separate out those perturbation effects, then that's great because it makes your perturbation modeling much easier, right? And it requires the latent space to capture very subtle phenotypic effects, especially in genetic perturbations, because those perturbation effects tend to be smaller than in drug perturbations. And so we call this the intrinsic evaluation of the embedding space, is how well does that embedding space separate different treatment groups? And it kind of measures the information content of the embeddings. And so we've plotted this on the y-axis, and the dotted line here is basically saying if I just train this classifier on highly varying genes or the raw expression space, how well does it separate by perturbation effect? And then on the x-axis, we have what we call the extrinsic evaluation of the cell embedding. And that basically measures how well is that separation maintained in predicted embeddings. So if I train a state transition model on that embedding space, how well does the simulated perturb-seq separate according to perturbation label, using the same classifier that was trained on the real data though. So we like to think about this extrinsic evaluation as how learnable is your embedding space. And so we found this is a pretty exciting capability of state in that it improves both the intrinsic and extrinsic performance of training perturbation models, especially in the genetic perturbation case. So we find this as like an exciting capability of denoising perturb-seq data to a degree. We also tried to use this to help our transfer learning. So the way we ended up presenting this in the paper is we trained a model on Tahoe 100 million. So this is on drug perturbations. And then we threw away the perturbation encoder and took that same pre-trained model and trained it for genetic perturbations now. So we trained it on 3 out of the 4 cell lines in Replogle, for example, and evaluated on the 4th cell line with no data seen in the specific test context there. And so this is like not only data set transfer, but it's also modality transfer of this perturbation. And you see that using the state embedding model improves over using the highly varying genes in every single case across all of these data sets here. And if you think about why, it's a really difficult task to go across modalities and across data sets. In fact, the going across modalities part, you would expect only to work with an embedding model that has some knowledge in its latent space about like different gene circuits and such. And so on this parse PBMC data set, we kind of dug a little bit deeper to look at all of the different metrics, and we see that using the embedding model improves over using just the highly varying gene representation, specifically in this held out context 0-shot setting. And then lastly, I'll go a little bit faster here through this last section. We used the results of the trained model to identify interesting cases to highlight for cell type specific effects. And so what we did was we plotted the perturb mean on the x-axis in the right direction and the context mean on the left axis. And if you improve over both of these baselines, kind of like we talked about earlier, it's evidence that the model is learning some cell type specific effect. And so we wanted to probe a little bit deeper and see, was the model able to identify any sort of interesting effect? Out of the top 2 ranked perturbations, one was an FDA-approved drug for BRAF mutant melanoma called Trametinib. And it just so happened that one of our test cell lines, C32, is a known BRAF mutant melanoma line. And so that seemed like a good candidate to kind of look for cell type specific effects and see if we were able to capture anything interesting that the mean baselines were not able to. When we look at the p-value distributions, we see that state is better calibrated than either of the mean baselines. This is just a plotting artifact, but actually what we see here is that these are all predicted to be significant. And typically the mean baselines will predict every single gene to be differentially expressed. Whereas the calibration of the p-values from the model outputs from state are more well, I guess, matching. We then, for each of the cell lines in the test set, so the test set that we chose for the Tahoe data set, we kind of put aside 5 cell lines and then right before the preprint evaluated on them and kind of ran the entire process on them. So for these 5 cell lines, what we did was we sorted by differentially expressed genes for each cell line. So that's how you get these clusters of, we sort by FDR for the most differentially expressed. And that leads to these cell type specific clusters of different genes. And so we plotted how well is state able to recover these specific DEGs as compared to like the perturb mean or the context mean baselines. And then we also summarize this in this histogram over here. So what is that overlap in predicted DEGs versus observed DEGs for state versus other models? And across all of these, we're able to see that state recovers many more of the true DEGs. I shouldn't say true, I should say observed DEGs. And in the C32 cell line, we're able to see the effect of Trametinib. We're able to capture a lot of these cell type specific effects that the mean baselines are not able to capture. And we're also able to capture some of these interesting non-cell type specific effects here as well. And so in summary, we find that state transition learns treatment effects across sets of cells and enables the model to resolve unannotated biological heterogeneity, improving perturbation response prediction in many settings, especially when you scale the data. And we find that state embedding model learns general cell representations and can reduce noise when training state transition across data sets and across modalities. And there's several future directions for the work here. One is that I guess as GenBio would know, it's never ideal to train so many models and then kind of plug and play them without training them end to end. And so a unified model that could combine state embedding and state transition would likely improve response prediction. Maybe in this novel context as well, like maybe that's the unlock to do that. Also adding in a natural language interface. This kind of helps because natural language has a lot of context on perturbations and different gene-to-gene relationships. And it would also be a nice way for biologists to start interfacing with the state model. And then lastly, enabling generalizations across perturbations and not just contexts. So this is bringing back the gears task of perturbation generalization, but also for unseen contexts. And by the way, this last task here is actually what ARC is asking folks to do for the virtual cell challenge. So it's quite a challenging task and more difficult than what we outlined in the state preprint, but I think it's possible with future advances in the methods. And so then just wanted to say thanks again to collaborators. Thanks to Caleb for inviting me on to speak. It was really fun to chat.
Awesome. Thank you so much, Abhi.
Thank you so much.
So I have one question from the chat that I think is going to be a good note to end on. But Shaheen, did you have any burning questions you wanted to ask before that?
No, most of them we have touched on. Yeah.
Okay, great. We're getting lots of applause in the chat. So great presentation, Abhi.
Thank
So
you.
one question that I'm sure is on everybody's mind who's developing these models, and that state seemed to outline very clearly. Why are genetic perturbations so bad? They seem intuitively like they should be much purer than small molecule effects. We know the target. Why are they so bad? And, how are we going to improve? Are they even worth, focusing on? What's your thoughts on this, your experience from state?
Yeah, I think it's a twofold, probably many morefold problem, but I'll maybe touch on two folds that we're thinking about. The first is the scale of the data, right? If you go back to the first slide, the replogal data set that we train on is around 500,000 cells after some QC filtering of on-target knockdown efficiency and such. And it's just it's like 2 or 3 orders of magnitude smaller than the Tahoe 100 million data set. So the first is that it's just expensive to generate like really deeply sequenced genetic perturbation data sets across a huge number of cell lines or contexts. I think the second thing is that genetic perturbation effects are just more subtle in general. First of all, you don't even know all the time if the applied effect was actually or the intended effect was actually applied in the cell. Whereas in a drug perturbation case, I mean, it just kind of wreaks havoc in the cell, right? So it's definitely being delivered to the cell. But even beyond that, a lot of times perturbations may not do anything, and that can also confuse models. I mean, it's not always trivial to learn the identity function. What I think is the promise of genetic perturbations is that it seems a much more tractable space to learn, right? There's only so many genes you can knock out in the eukaryotic cell. And so if you can actually generalize to multiple perturbations at once, again, not super, I say that like it's an easy task. It's not an easy task, but it seems more tractable to me than learning to generalize to all of chemical space, for example. And so there are actually translational modalities for this as well, like, right, like CRISPR overactivation. That's basically mRNA therapeutics that you put into the cell and then it would express that. And so there are companies that are doing overexpression, for example, as a modality. I think the data is just not there, and the phenotypic effects for the model to learn are just much more subtle. Both of those make it super difficult, I think, to improve performance.
Okay, great. I hope that that helps everybody who might be tuning in working on these models. This is a great note to end on. Thanks so much, Abhi. This is a great model and a great paper, and you're a great speaker. So thanks for being here. And we'll see everybody next time. Okay, take care. Bye-bye.
Thanks. Great chatting. Thanks.